All right, so what we're going to look at now is a respirometer. We are going to be doing cell respiration. So I am going to show setting up a respirometer. I have, through other friends, have found I have found that using a syringe and a pipette and some tubing is by far a much better method to do cell respiration because you do not have to submerge it in water. It is not like the old school way where it is going to have to sit in the tub of water and all of that. So what we're gonna have, this respirometer will have, this is a 60 ml syringe. This is a 2 ml pipette. You want a 2 ml pipette. You do not want a 1 ml pipette because a lot of these plant materials will go through 1 milliliter of oxygen in the 30 minutes and you would then have a problem. So you need a 2 ml pipette and the tubing, this is normal tubing, aquarium tubing, it's all one size. So you need those things. Then you need to have just regular old cotton balls. Then you need to have non-absorbent cotton balls. So you can get non-absorbent cotton balls through the biological companies, but if it is a problem finding it, I have used in the past at different APSIs, I have used polyfill, which is the normal stuffing that you would stuff a pillow or an animal with, and it is non-absorbent and it will work just fine. It's rayon based. And the last thing you're gonna need is the potassium hydroxide. The reason why we're using potassium hydroxide is the whole idea is we're looking at cell respiration. We want to see that the peas or the beans are breathing, that they are consuming oxygen. Well, as they are consuming oxygen, they are also expelling CO2. If we did not have something in our respirometer to capture the CO2, then we would not be able to see the movement of oxygen going in. So what we're gonna do is, and following the instructions, we're going to take, and I'm going to do, I have both here just so that y'all could see, because with my students, I would for sure have the different ones. This is 48 hours for pinto beans. This is 48 hours for black-eyed peas. This is 24 hours for pinto beans, excuse me, and 24 hours for black-eyed peas. Normally with the students, what I would have them do is I'd have them run the first trial, and that would be with, let's say, the beans of 24 hours and the beans of 48 hours. After that, they are then free to switch and do other ones. Normally I have other beans as well. Mung beans are really cool. They're really little, but they respire a lot. So I like to use those as well. But it's nice for the students to do the first, which is your baseline. So this would be like the 24 and the 24, or the 24 of the black-eyed and the 48 of the black-eyed or of the pinto. And then for the second round, to then switch up and just do whatever they would like. If you have the availability, or you can get bigger syringes, you could go ahead and also do some animals. You could do some pill bugs, you could do some mealworms, you could even do grasshoppers. Grasshoppers, keeping in mind they like to hop. So I've done it with teachers, not with students, because they go everywhere. So go ahead and cut.